Hi, I'm Ikko Kimura, a postdoctoral researcher at DLCMR. Today, I'm going to talk about my recently published study that looks into whether the microstructure changes in the first place after repeated transcranial magnetic stimulation, or rTMS. Whether the microstructure changes rapidly or not after rTMS is a kind of a controversial topic. And I try to answer these questions by using a quadripulse stimulation, or QPS. Well, QPS is one of the rTMS protocol whose after effect is relatively strong and robust compared to other protocol of the rTMS. And as a result, we found no significant changes in the microstructures, and we believe that this result is important to clarify the underlying mechanisms of the rTMS. Okay, so here is the contents. After showing the backgrounds about the general microstructural plasticity and the quadripulse missions, I go over the methods and results that has been recently published in the Imaging Neuroscience with discussions. And finally, I'm going to provide a few potential future directions of this study. Okay, so let's start with the backgrounds. We all know that if we activate the neurons repetitively, that can change the microstructures. For example, this figure shows the dendrites of the rat hippocampus. And if we activate the neurons repetitively for 1 hour, that connects the dendrites, that elongates the dendritic spines, as you can see. And this figure shows the astrocytes after the 2-hour water weight task in rat hippocampus. And as you can see, compared to the control conditions, the astrocyte size got bigger after the behavior task. And not only the gray matter, one study shows that there are changes in the white matter near the just below the stimulated regions. And when it comes to the human studies, of course, we cannot assess the microstructural property directly. This is because we cannot cut the human brain, the living human brain. So we need the non-invasive ways to assess the microstructures. And diffusion MRI, or DMRI, is one of the ways to assess the microstructural property. Well, diffusion MRI evaluates the diffusibility of the water along multiple directions. And we can see the property of the microstructures by seeing how the diffusibility of the water is restricted by the surrounding microstructures. Here I talk about the microstructures, but like neural cells, or the axons or myelin, or the glial cells like astrocytes or microglial cells. And doing a model fitting to the diffusibility of the water on each voxel, we can quantify the some perspectives of the microstructure property. For example, fractional anisotropy calculates the diffusibility of the water along main orientations of the fibers compared to its orthogonal directions, and that basically reflects the axon density or myelinations. And in the meanwhile, mean diffusibility literally calculates the diffusibility, average of the diffusibility of the water across all the directions. And this basically means the spasticity of the cell. Well, I'm going to use this two terms frequently after this slide, so please remember these two words. So again, fractional isotropy, high FA basically means high axonal density or high myelinations, while low MD basically means high cell density. Okay, and by using the diffusion MRI, several studies clarify that the microstructure can dynamically change in the human adults. Like this study looked into the FA changes after the juggling, and they showed that the FA increased after the juggling in the white matter that is related to the visual spatial processing. And this study looked into the FA changes after the neurofeedback to change the degree of the co-activations between the two regions. So neurofeedback to increase the degree of the co-activations between the bilateral sensory motor area, increase the fractional anisotropy in the white matter that connects these two brain regions, while the neurofeedback to decrease the degree of the co-activations in turn decrease the fractional anisotropy in the same regions 1 day after the neurofeedback. And more surprisingly, like a microstructure can change in much faster pace than we have ever thought. Like 2 hours of the special memory task or the 45 minutes of the piano training is enough to change the micros, to decrease the mean diffusivity in the task-related gray matter. And what is surprising is that such decrease of the value was observed right after the single sessions of these behavioral tasks. But behavioral training rapidly activates the widespread brain networks. And we still don't know whether the rapid activation of just the single brain regions also changes the microstructure in such a fast pace or not. And one way to stimulate the single brain region repetitively is rapid transcranial magnetic stimulation, or rTMS. Well, I guess most of you know about the rTMS, but for those who don't know rTMS uses a device like this, and we put the core on the head near the targeted regions to stimulate the targeted regions. And we can repetitively stimulate the targeted regions by repetitively emitting the electric pulse from the main device to the core through electromagnetic inductions. And several studies already look into whether the rTMS can rapidly change the microstructure or not, but as I said, the result is inconsistent across studies. For instance, one single session of the 1 Hz rTMS decreased the mean diffusivity in the stimulated regions, but other studies showed that near the same protocol of the rTMS actually increased diffusivity, both in the stimulated and non-stimulated regions. And on the other hand, 2 other studies showed that there were no actually any significant changes in mean diffusivity. And of course, there are several possibilities to explain these inconsistencies. But one potential reason would be that the rTMS after effect is highly variable across subjects. And by the way, the output of the paramotor cortex can be measured by the response from the single pulse of the TMS over paramotor cortex, or M1. And we can see the response by taking a electromyography, which is called a motor evoked potential, or MEP. And by taking a MEP after the excited protocol of the rTMS, which is expected to increase the MEP, in this case, the value is expected to be higher than 1, while few participants increase the MEP, but as you can see, other participants actually decrease the MEP. And taking the average of the MEP amplitude change, actually there were no changes, as you can see in this bold black line. So we need the rTMS protocol that induce robust and strong after effect. And I believe that the quadripulse stimulations, or QPS, is one of the promising rTMS protocol. Unlike the previous studies that use the conventional rTMS, which stimulates the brain with fixed frequency, quadripulse stimulation is one of the patterned rTMS protocol that stimulates the brain with 4 pulses every 5 seconds for 30 minutes. And the QPS with short interval as short as 5 milliseconds increases the brain activity of the stimulated regions, while the QPS with long interval as long as 15 milliseconds decreases the brain activity. And another protocol of the patterned rTMS include the famous theta pulse stimulations, or TPS. And previous study already compared the, directly compared the effect of the theta pulse stimulations and QPS. This pie chart shows the rate of the respondent after the QPS or TPS. And so here, responder means whether the MEP change was expected after the rTMS over left and 1. And upper panel shows the responder rate of the TPS, while lower panel shows the responder rate of the QPS. And the left panel shows the comparison within the rTMS protocol that is expected to increase MEP, while the right panel shows the comparisons between the inhibitory protocols of the rTMS. And as you can see, the rate of the responder, which is in the dark shaded regions, was higher in the QPS compared to the theta pulse stimulations in each time course and across protocols. And one noteworthy thing is while the responder rate is around 60% or 70% in TPS, the rate of the responder in the QPS goes as high as around 90% or even 100%. But however, actually the brain stimulator that used in this previous study has not been solved anymore. And if we start the new QPS project in the lab, we may have to use a newly developed stimulator like this. But we still don't know whether the effect induced by this newly developed stimulator is as strong and robust as in the previous studies. So in this study, I first confirmed its after effect. So again, I performed the QPS over left paramotor cortex and assessed the MEP amplitude change. And the left panel shows the MEP amplitude change after excited protocol of the QPS, while right panel shows the inhibitory protocol of the QPS. And thick black line shows the mean MEP amplitude change across participants, while other line shows the individual MEP amplitude change. And as you can see, QPS5 increased the MEP amplitude in all the participants, and the mean MEP amplitude change was within the range as expected from the previous studies. And QPS50 also decreased the MEP amplitude change, MEP amplitude, and the MEP amplitude change was within the range that is assumed by the previous studies. So here I confirm that I can also use this newly developed QPS estimator, and using this new QPS estimator, I first ask whether the FA or MD value change in the first phase after the repeated activation of single brain region by QPS. And I also ask whether such a microstructure changes is related to the functional change after QPS. And here I hypothesize that the FA or MD value change as in the behavior studies, behavior training studies after QPS 5 and QPS 50. But since the functional change after the QPS 5 and QPS 50 is in the different directions, so I thought that microstructure change is also goes in the different directions between the QPS 5 and QPS 50. And I also hypothesized that the functional change is related to the microstructural change after QPS. Okay, so here I want to see whether the microstructure property change right after rTMS or not. Okay, so here is the overview of the experiment. I recruited 16 right-handed healthy adults, and the experiment consists of the 3 days. And on day 1, we took task fMRI during finger tapping task to localize the left palmomotor cortex. And after that, we did the structural MRI scans to do the group analysis. And on days 2 and 3, we have performed the QPS 5 or QPS 50 on separate days. And QPS was performed over left palmomotor cortex. And before and after QPS, resting state fMRI and diffusion MRI were obtained to assess the functional changes and microstructural changes, respectively. And from diffusion MRI, I calculated the FA and MD value, and FA and MD maps was analyzed in the separate structures to have a better alignment across subjects. So in the white matter, I used the tract-based spatial statistics, and on cerebral cortex, I used the surface-based analysis combining multimodal surface matching. Well, and the multimodal surface matching uses the information of the resting state fMRI or myelin mapping to have a better alignment across subjects compared to the surface-based analysis alone. And as for the subcortical structures, I used the voxel-based analysis. And as I said, functional effect was assessed by the resting state fMRI. And more specifically, I used the functional connectivity changes between the stimulated regions and its control lateral regions. This is because previous studies showed that the functional connectivity changes in these regions was related to the QPS effect over left M1 or MEP amplitude change. So in this study, they showed that the functional connectivity was decreased in the QPS 5 while increased in the after QPS 50. And the functional connectivity change and MEP amplitude change was significantly and negatively correlated in both QPS 5 and QPS 50 conditions. So here, using the diffusion MRI, I first compared the FA and MD values before and after QPS 5 or QPS 50. And using the resting state fMRI, I looked into the relationship between the functional changes and microstructural changes. Okay, so here comes the result. Well, as I said, there were no significant changes in most regions. And this figure shows the base factor in comparison of the FA value before and after QPS. And the blue shaded regions shows that the base factor is smaller than 1, which indicates that the data relatively supports the hypothesis that there are no changes. And as you can see, the base factor was smaller than 1 in most regions, both after QPS 5 and QPS 50. And the classical hypothetical testing also showed that there were no significant changes in any regions, either after QPS 5 or QPS 50. And that is the same is true for the mean diffusivity on the cerebral cortex. As you can see, most regions are shaded in blue. And also the same thing is true for the FA or MD value in the white matter. And only significant changes I could observe was the significant decrease of the mean diffusivity in the left cerebellum after QPS 5. But I think we still need to carefully interpret this result because voxel-based analysis that use on this structure can be sensitive to the registration errors compared to the surface-based analysis that I use on the cerebral cortex. And one thing to note is that the left cerebellum is not directly connected, not in most cases connected to the stimulated regions, because left cerebellum is mainly connected to the right cerebral cortex. So it is less likely to be directly affected by the stimulated regions. So I would say that this is a kind of a false positive. And as for the functional changes, this figure shows the functional connectivity change after QPS 5 and QPS 50. And as you can see, the QPS 5 decreased the functional connectivity, while the QPS 50 did not show any significant changes. And the functional connectivity change was not significantly correlated with the change of the microstructure property in any regions, which implies that microstructure changes did not occur even in the participants who had more functional changes. To summarize the result, well, we found that microstructure property was not changed in most of the regions, either after QPS 5 or QPS 50. And this supports the 2 out of 4 previous studies that did not show any significant changes in mean diffusivity after rTMS. And together with these 2 studies and our studies, I would say microstructure would not change in a fast pace after rTMS, which is quite reasonable when we think about the time course of the synaptic plasticity. More specifically, morphological events would occur after the electrophysiological events or the biochemical events. And that was supposed to take a few hours or days. But as I said in the introductions, several studies showed that the behavioral trainings can change the microstructure rapidly. And the difference of the these studies and our studies is it would be due to, I think, would be due to the differences in induced activity between the behavior training and rTMS. For instance, behavior training induces the activation intrinsically, while QPS induces extrinsic activations. And while behavior training activates the widespread brain network, QPS activates the brain more locally. And while the behavior training, like even within the same area, while the behavior training activates both superficial and deep layers, QPS activates the, or substantial data rTMS activates mainly the axon terminal of the superficial pyramidal neurons. Okay. So here we showed that there were no rapid changes in the microstructure after QPS. And with the last 10 minutes, I would like to show a few potential directions, future directions of this study. So here we did not show that there were no microstructure changes right after the rTMS. Then does this mean that the single session of the rTMS is not enough to induce microstructural plasticity? Well, I wouldn't say so, because several studies showed that the single session of the rTMS actually changed the microstructure. For example, in vitro, 10 Hz of the repetitive magnetic stimulations increase the dendritic spine size a few hours after the stimulations, while the other protocol, the same protocol of the rMS morphometrically change the inhibitory interneurons 3 hours after the stimulations. And also another study showed that the excitatory protocol of the rTMS decreased the spine density in mouse after 45 hours the stimulations. And actually, like other type of the neuromodulations was also shown to change the microstructures by single sessions. For example, the neurofeedback decreased the fractional isotopy 1 hour after the training in the widespread white matter. And the other protocol of the neurofeedback, as I showed in the introductions, bidirectionally changed the fractional isotopy 1 day after the training. So, as in this study, there is still a possibility that the microstructure can change 1 hour after the QPS. And one of the advantages of the QPS is that the after effect lasts long. For example, after effect of the QPS5 lasts long as a few hours, which is quite unexpected, considering the other protocol of the rTMS. And also QPS50's after effect can last more than 1 hour, at least. So it will be really interesting to see whether the microstructure change several hours after the QPS or QPS5. And another interesting future directions would be to assess the microstructures multimodally. This is because when we talk about the microstructures, that can mean a variety of the things. For example, this can mean the changes in the oligodendrocytes and the myelinations, but that can mean the morphometric change in the axons, or the growth of the glial cells, or vessel structure. So Well, I still believe that the diffusion MRI is sensitive enough to see the microstructure changes. We still need to clarify which type of the microstructure change after the neuromodulations or the behavior training. So maybe myelin changes or axon changes or not. And with the last few slides, I'm going to briefly introduce the technical advancements of the non-invasive assessment of some type of the microstructures that I used previously. Well, first, we can assess the myelin content non-invasively with MRI. And one way to do that is to use the T1-weighted and T2-weighted data. This is because T1-weighted data is proportional to the myelin contrast, while T2-weighted data is proportional to the inverse of the myelin contrast. So taking a square root of the T1-weighted data divided by T2-weighted data, we expect to cancel the bias to extract the myelin contrast. And this study looked into the laminar profile of the macaque brain and on the several regions. And this blue line shows the laminar profile of the myelin level with histology, while this orange line shows the laminar profile level with this myelin mapping method, or MRI method. And x-axis shows how deep the profile is, going from the deeper layer to the superficial layer. And as you can see, this MRI method successfully replicated the laminar profile level with histology. So in the near future, there's a potential to see whether the superficial or deeper regions of the myelin changes after the stimulations. And we can also assess the vessel changes after with MRI. And this can be done by using the contrast of the iron with MRI, which we call the R2 star, before and after injecting felmoxyl, which is the iron-rich content. And this figure shows the R2 star map of the macaque brain after injecting felmoxyl, while this map shows before felmoxyl. And as you can see, the felmoxyl enhanced the large vessel structure. And subtracting the R2 star map after the felmoxyl with that before felmoxyl, we can derive the vessel density, which we called delta R2 star map. And doing a special filtering, we can also extract the structure of the large vessels like this. And from here, we can see we may be able to see the diameter of the vessel or the vessel structures. So again, maybe in the near future, maybe we may be able to see the vessel density changes or vessel diameter changes after the behavior training or the neuromodulations. To sum up, here, it would be really appealing for me to assess the microstructural changes with multiple time points using multimodal MRIs. And it will also interesting to assess the microstructure changes, not only the rTMS, but also the other type of the protocols, other type of the neuromodulation protocols, like transcranial electric stimulations or TDCS or TSCS, and transcranial ultrasound simulations. To clarify the advantage or disadvantage of these type of the protocols. Okay, so to summarize my talk, I'm using quadripulse stimulations, which induces strong and robust after effect, which showed that the microstructural property did not rapidly changes in most regions after both excitatory and inhibitory protocol of the QPS. And from these results, we speculate that such an extrinsic rapid activation of just a single brain lesion is not enough for microstructure to change rapidly. And as a future directions, it will be very interesting to assess the microstructure changes after each type of the neuromodulations using multimodal MRI at multiple time points. Well, thank you for your attention, and I would be happy to have any questions. Thank you.